need some space. Aesthetically, it must be accessible, this region, to be able to have transcription activated there. So euchromatin are typically the active genes. Heterochromatin, if your gene is located in the heterochromatin part of our genome, then you will have no expression of that gene. And it's so compact that really a lot of that DNA is there. So by compacting it so much by the heterochromatin state, although it would seem, you would have the impression here that there is more euchromatin than heterochromatin, but in reality that's not the case. If you would look how much of our genes are in the heterochromatin states in every single cell, then that would be around a bit more than 90% of the genome is in the inactive state. Only 10% of the genome per cell is in the active state, and that can vary between the cells. Between tissues, heterochromatin versus euchromatin state will be different, although there are a few parts which are constitutively in heterochromatin state as well. So, in the heterochromatin, some parts are constitutively heterochromatin. Some parts of our chromosomes, mainly at the centromere and the telomere regions, you have a very strong condensation. You have continuous heterochromatin state there. Also some repetitive sequences, like transposable elements, but also around the centromeres you have a lot of repetitive sequences. They are constitutively in heterochromatin state so that they stay silent. So that they do not, the transposable element, we don't want them to jump around in the genome. That would cause genomic instability. So that is why it's so very important to keep this in heterochromatin in silent state. Also, the telomere ends are typically constitutive heterochromatin. However, there are also parts of the genome which are facultative heterochromatin. That means that they can switch between the states. So they are heterochromatin at a certain moment in development or in a regular condition, but then upon a certain trigger, like, I don't know, a part of a certain molecule comes in the cell, and then that switches. It goes into euchromatin state. So specific regions that are involved in differentiation, that respond to the environment, those regions are in facultative heterochromatin so that they can be activated when needed. So that chromatin, how does it look? If you would, this is an electron micrograph picture of the 30 nanometer fiber. Now this is only observable, this fiber, in an experimental condition. So in really, if you isolate the chromatin from the cell, in reality that state does not exist in the cell, but that's the state that we can see with an electron microscope, so that is why it's always represented like that. This has of course been instrumental to be able to study chromatin. So the 30 nanometer fiber that we experimentally can isolate is composed of nucleosomes. Nucleosomes are DNA wound around histones. And histones are octameres. They consist of 8 subunits. 8 subunits around which that DNA is then wound. Now DNA has a negative charge because of the phosphate groups. Histones have a positive charge. So they loosely interact with each other. It's not very strong, this connection, but there is a connection. They tend to attract each other by their opposite charges. And then if you see here the DNA wound around these 8 subunits of the histone. Here you see it again, and if you would look at these things here, these are the histone tails. These things that protrude out of that core, let's say, are the N-terminal tails of those histones. And the histones are proteins. They have an N-terminus and a C-terminus. This is the N-terminus of those proteins. They come out. So they are protruding a little bit from that core. What else can I say about that? Well, maybe that this is the DNA. There's a double helix wound around one histone. And that is typically around 150 bases. And I say around because it varies between species. But that's the rule of thumb, around 150 bases is around one histone. And that histone consists of those 8 subunits. The subunits are called H2A, histone 2A, H2B, histone 2B, histone 3, H3, and histone 4. We will talk later about histone 1. Histone 1 also exists. They make dimers. So H2A and H2B make a dimer. H3 and H4 make a dimer. Then those two, again, pair with each other. 2 dimers make a tetramer. These two, again, 2 dimers make a tetramer. Then you have 2 tetramers that compose the octamer. Happens extremely often. With proteins, they often form complexes with each other. So proteins typically don't work alone, but in big groups together that aggregate with each other and form this huge structure here. It's quite big in comparison with the single protein. Now histone 1 also exists, and histone 1 is in fact the lock. I call it the lock because it connects the nucleosomes tightly together. So it will strengthen the association between the histones and DNA. So between the other subunits and the DNA, histone 1 stabilizes that interaction. So it's it's this thing here that locks everything together. The more H1 you have, the more condensed your chromatin is. And to keep that connection very close, very compact, histone 1 is very important. So the more you can really detect also the heterochromatin versus euchromatin states of the chromatin just by checking how much histone 1 do I have, and the more you have, the more likely it's heterochromatin. Now, these N-terminal tails that I talked about, I specifically indicated them, the ones that recruit out of that nucleosome core, are post-translationally modified. And what is post-translational modification? Well, post-translational modification means the equivalent process of changing proteins after biosynthesis. I hope you all remember how proteins are being made, and transcription and then translation. After that translation, those proteins that are built are being modified by tiny changes. Tiny changes on specific amino acids, on specific residues. Some little molecules are added there, some small groups like acetyl groups, methyl groups. Phosphorylation is also an option. Ubiquitin can be added, the sumo group can be added. Then there are some things that look a bit like ubiquitin but are not exactly the same. Glycosylation, and so on and so on. Also, sometimes a protein is just cleaved in half, and the 2 halves each have their own function. So all those things, those changes that happen after synthesis of the protein, after the translation, we call that in general PTMs, or post-translational modifications. And some of those post-translational modifications are, need to be catalyzed by enzymes, and others not. For example, some redox reactions or oxidation of proteins happen spontaneously without enzymes. Also nitrosylation happens spontaneously, but others require enzymes. The ones that we are going to talk about today require enzymes. So I'm going to each time specify which enzymes are. So histone modifications are N-terminal post-translational modifications. Post-translational modification on the N-terminus of the histones. And there are quite a lot, more and more every, let's say, every month a new one is discovered. Quite a lot of changes happen on those histones. The most commonly observed ones, and the ones that we know most about, are here shown in big letters: acetylation and methylation. But there are more. Phosphorylation, ubiquitination, and so on and so on and so on. Hydroxylation, simulation have all been described. And one of our guest lecturers that will present his work in May is specifically working on histone modifications, so he will then explain for sure a lot more about that and the world of the histones. Which he is calling that histonomics because he looks at genome-wide histone modifications and their effects and also their evolution. But that is for the guest lecture. Histone 3, histone 4, H2A, H2B, H1, they can all be internally modified. And here, all the ones that have been described are just shown here as an overview for you, so that you see how many there are, how many different ones there are, but you will very often see that it's methylated or acylated. Those are the ones that we find more prominently, more often on histone tails than the others. So that is why those two have, of course, been studied in more detail than the other ones. So the first one is histone acetylation. Histone acetylation is typically happening on lysines. On lysines, in the N-terminus, some of the amino acids there are lysine, like at the 9th position, that is a lysine. But also, arginine can be acetylated. We will see some examples of that in the next slide. But lysine, when it is acetylated, you need, of course, an acetyl donor, and that's acetyl coenzyme A. And you need an enzyme to catalyze this reaction, that's histone acetyl transferase. This will put the acetyl group here at the epsilon position, so at the NH3+, which is then neutralized. So there's no longer, lysine is a positively charged amino acid, but when acetyl is added, it's no longer positively charged. So the charge is neutralized by this change, by this acetylation. And I said that the charge was very important for the interaction with DNA. I said histones are positive, DNA is negative. But here you neutralize part of that positive charge of the histones. Doing so, you weaken the interaction. So the interaction with the DNA is weakened by those acetyl groups on the N-terminal of the histones. So the more histone acetylation there is, the weaker the interaction between DNA and the histones, leading to open, more euchromatin state activation of transcription. So we see a clear correlation between more histone acetylation and transcription. This correlation is quite, universal. So we see it very often that acetylation activates gene expression. I will also tell you that for the other histone marks, it's not that clear, this correlation. Acetylation is the one where we really see a very clear interaction. So the histone acetylation is a very clear association with activated regions. So in euchromatin, you have a lot of histone acetylation. So here, the closed chromatin, if you would add a histone acetyl transferase acetylating the histones, you change that state, you push it towards the open chromatin, the euchromatin state, so that genes can be activated. And the other way around, of course, also happens. To go back to the heterochromatin state, you can deacetylate the histone. So in the cell, there are enzymes that do that, and those enzymes are called histone deacetylases. They will remove acetyl groups from the N-terminal histone. So that typically leads to transcriptional activation, while this direction leads to gene silencing. So the more complex one is histone methylation. Histone methylation is mediated by the enzymes I've shown there on top, histone methyltransferase, and the removal of methyl groups from the amino acids is mediated by histone demethylase. Now, sometimes, I don't know if you know that we typically abbreviate proteins with between 3 and 5 letters. We prefer to have 3 letters, like HMT, or later I will talk about IGF, HDM. But also some people tend to not keep to the consensus. Let's say, well, they do stay with the same 3-letter abbreviation, but then suddenly they call it, instead of histone methyltransferase, KMT, and that stands for lysin methyltransferase. Right? The K, the 1-letter code of the amino acids, K stands for lysin. So some people, if it's specifically a lysin methyltransferase, they call those enzymes KMT and KDM for the lysin demethylase. So that's a subgroup of the histone methyltransferase that specifically works only on lysin. Because not only lysin can be methylated, sometimes also arginine is methylated. So here you see what happens if lysin is methylated, and you will see that it's not necessarily one methyl group. It can be 1, it can be 2, or it can be 3. So monomethyl lysin exists, but also dimethyl lysin and trimethyl lysin. All mediated by histone methyltransferase. So each time for the transfer you need an enzymatic reaction adding a methyl group. At arginine, there is even different potential methyl groups, different options are there, different stereomers. There's monomethyl arginine, but then there is also dimethyl arginine, which is symmetric, or asymmetric dimethyl arginine. So here, depending on where the methyl groups are put on the structure. Now, adding a methyl group does not have an effect on the charge of the histone, like what acetylation did have. So here there is no effect on the histone charge. What we do know about this versus acetylation is that methylation is more stable. Acetylation is easily removed. Methylation is a bit more strong, stays long, let's say. And what is extremely important to realize is that its effect on expression is variable. I read it every year on the exam that people say histone methylation silences the gene. It's not true. DNA methylation is typically associated and then only in the promoter leads to silence. Histone methylation, I cannot predict what will happen with expression of that gene. There's no clear correlation because it depends really on which residue of the histone tail we have the methylation, how many, is there 1 or 2 or 3, and also even on organism. So it varies between, for example, Drosophila versus zebrafish, and it will already be different, the effect of methylation. So this is a very, hard one to study because you cannot predict what it will do. But we do see that a lot of those histone tails are methylated, so it must mean something. Histone methyltransferases typically contain the so-called set domain, and that set domain, we will talk about CEF39H1, a human histone methyltransferase, in one of the next slides. You might, I'm now wondering if it's today or next week that it's probably next week that this protein will pop up again. That's why I specifically put it on this slide. CEF39H1 is a human histone methyltransferase, and it can do these 3 reactions: monomethylate, demethylate, and then trimethylate. Histone 3 lysin 9. So the histone code is typically abbreviated like this: H3K9 means on histone 3, the subunit H3, the lysin K at position 9. And then if it's methylated, it puts as a superscript methyl, ME, or AC, acetyl, for acetylation. This is what you see here. I'm using this here for showing. It's very often we put it as a superscript, but my superscript seemed to have disappeared here in my slide. But you can also put it just behind it. That still, but you will often see that the AC is shown here as a superscript. H3 lysine 9 acetylation. H3 lysine 9 trimethylation. H3 lysine 27 trimethylation. H3 lysine 4 trimethylation, and so on. So there are quite a lot of lysines, lots of Ks in the N-terminus of those of H3, and all of them can be modified, and all of them can have a different results on gene expression. Now, these are the common rules, let's say, but there are always exceptions to many of those rules. For acetylation, we have the strongest association. If there is histone 3 lysine 9 acetylation in the promoter, the gene is typically active. But for methylation, you already see different options here. H3K9 trimethylation in the promoter, and these two are of course exchangeable. It's either acetylation or methylation at that same position. If it's acetylation, it's active. If it's methylated, it's typically silent. But then at those other positions, it really depends not only on the location in the N-terminus of the histone, but also the location in the gene. So here it was the promoter, but here we know that H3K27 trimethylation in the gene body is typically seen in genes that are silenced. H3 lysine 4 trimethylation in the promoter is seen in genes that are potentially active, not yet active, but could be active. So genes that are ready to be active, we call them poised for activation. That's a mark that's typically associated with genes that are like waiting for a signal and then they will be quickly activated. H3K13/6 trimethylation, also often seen in exon regions of genes that are. All these associations, it's all correlation mainly. Yeah? Now, of course, we can explain for acetylation why those genes are more active, but for the methylation, it's way more difficult because they do not have an effect on the charge of the histone, so that interaction effect that you had with acetylation, while you neutralize the positive charge of the histone, is not happening for methylation. So there, it's harder to predict what will happen. So we just have to look how often do we see this mark on the histones, and do we see an association with activity of the gene. Now, I talked about methylation, I talked about acetylation, and another quite commonly found mark is phosphorylation, adding the phosphate group, typically at the site setting, tyrosine, for example. And that is mediated by protein kinases that add a phosphate, or phosphatases that remove phosphate. So these enzymes that put the mark there or remove the mark are sometimes called also the writers and the erasers. So they write the histone code, they add a certain mark to the histone tail, and then we call them the writer, or they remove it, we call them the eraser. So here, histone acetyltransferases mediate acetylation, histone deacetylases remove acetyl group. For methylation, I told you, histone methyltransferase or lysine methyltransferase isn't specifically only one. Because you, of course, also have the demethylases, and also on arginine, methylation can happen, and then we call it arginine methyltransferases. And that person that invented the name did not work with the 3-letter code, but now with the 4-letter code, PRMT. And then you have proteins that recognize these marks, of course. You have proteins that are going to bind to chromatin and only do that when that mark is present, or when they are attracted, let's say, to regions with lots of methylation. This is histone modification binding proteins. readers, they read the histone code. They will try to go to certain regions, they will specifically bind to specific regions of the genome where there is a lot of acetylation, for example. And those proteins have a domain that interacts with those acetyl groups. For example, like the bromo domain that interacts with acylated histones. So bromo domain containing proteins are specifically binding to acylated histones. Phosphorylated histones are being read by the 14-3 proteins. And then here you have different classes of proteins that can interact with methylated histones. MBT proteins to the chromodomain PHP proteins. I put those things here on the slide so that if you would find them in your literature, if you're reading something, a manuscript that will provide you manuscripts for the assignment, you might come across some of these names so that at least you know, that's a protein that interacts with a modified histone. So this part you don't have to study, but this I would like you to know. I would like you, at least this is not so hard to remember which enzymes can drive and erase histone codes. The readers, that's just for your information. If you would come across them, that you know, this is one of those readers. So a chromodomain is, for example, a very typical domain that binds to a methylated H3. And one example of a protein that is well known to contain a chromodomain is heterochromatin protein 1, H1. Watch out, do not confuse that with H1. That's not the same. H1 is a histone, the lock. HP1 is another protein. And HP1 has a chromodomain and binds to constitutive heterochromatin. So at the telomeres, at the centromeres, you have a lot of HP1 bound to that region because of the methylated histone. So what all these modifications lead to something that we call the histone code. So each histone is modified in some sense, and it's a kind of a code that we can try to decipher. All these N-terminal histone modifications can have effects on the transcription of the genes that they are bound to. The histones are not the genes, but they are bound to specific regions of the genome, so they can have an effect there on the transcription of the connected DNA. But the histone code is not easy to decipher. And that is because the effects of those modifications are variable between organisms. They change between, but also are depending on the location and on the number of changes. Like for example, here, histone 3, lysine 4 can be monomethylated, demethylated, or trimethylated. Then, that's a quite easy one. In Drosophila, it's connected to euchromatin, also in mammals, and in Arabidopsis. So in plants, animals, and in insects, we know that this is typically found in euchromatin regions. Because this mark is the one that I talked about 3 slides ago on genes that are ready to be activated, poised for activation. So typically found this mark in euchromatin states. For acetylation, as I said, always in active genes, euchromatin. So some of them are not so difficult. But then you get the annoying ones, let's say, like histone 3 lysine 9 monomethylation is in heterochromatin, often, but not always. Then already something between brackets an exception to that rule. In mammals it's in euchromatin, and in Arabidopsis it's in heterochromatin that we find that one. This one as well, variable. This one as well, variable between the species. Phosphorylation. Euchromatin, typically found in a lot of phosphorylation at serine 10, In Drosophila euchromatin, mammals euchromatin, plants heterochromatin. So you see all those differences make it very hard to study and to predict based on just the histone marks on the histone code what will happen to those genes. And those modifications are also very dynamic. So acetylation, methylation, depending on how the cell is feeling that day. Let's say how the environment of the cell is. Is the cell stressed or is the cell healthy? Is the cell growing normally? Is it responding to something? Already the marks will change. And it can happen very fast. Those changes, of course, happen within a few minutes. And that is, of course, very important for our genome to be able to respond quickly to the environment. So changing the marks on the histones is a very easy way of changing from heterochromatin to euchromatin state in that type of the heterochromatin. So, of course, again, you don't have to study this because it doesn't make sense to study it. I'm sure there are already multiple exceptions to that. It's just an illustration of what I put that on the left, and that I find very important that you know that those modifications can have an effect on transcription, but that the effect depends on multiple things. On the type of modification, on their location, and even on the organism in which you are, the organism that you are studying. And moreover, they influence each other. So I already explained that it's of course exchangeable. This is an N-terminus of histone 3, or is it 4? 3, yeah. 3. You see here, the fourth lysine can be methylated. So this is H3K4, can be methylated. H3K9 can be methylated or acetylated. It's one or the other. So, depending on what it is, you will see other things happening. If it's acetylated, then SUF39, that's the one I was referring to a few slides ago, and I said, I think it's somewhere on one of my slides, it's here. It's a human histone 3 lysin-9 methyltransferase. It's blocked. So if you have acetylation here, you can of course inhibit, so the methyltransferase is inhibited. So it's either this one or the other one. But the same happens if there is on serine 10 a phosphate group. Again, SUF39 is inhibited. So these marks here below are all euchromatin related, while the ones on top that we see there are more heterochromatin related. If there is a methyl group here, H3K9 methyl, that attracts heterochromatin 1. And heterochromatin 1 leads to heterochromatinization of the DNA, of the chromatin where that histone is present. So you see lots of interactions here. Lots of interactions, and addition of one group can inhibit addition of another one or promote addition of another one. Those things are all described in multiple species. Again, not always following the same rules in every species. So in summary, the writers add the mark to the histone, they introduce the mark. The eraser removes the mark, and the readers interpret the mark. They interact with the mark and produce then something else, for example, attract some protein that is needed at that location. So we call all those marks the histone codes. They make part of the epigenome. So it's the layer on top of our DNA that controls gene expression. That epigenome is not only consisting of histone marks. Last week I already explained to you that DNA can also be methylated itself, mainly at the cytosine. 5-methylcytosine in our DNA is typically associated with suppression of genes when it is happening in the chromosome. However, if the methylation is in The coding region, that's associated with activity of the gene. So, yeah. Also there, no clear rule, although most people believe and will still say DNA methylation leads to silencing. There are already a lot of exceptions to that. In the promoter, yes, but if it's not in the promoter region, then the effects are not so often we see active genes in the coding region have lots of them. Good. So that was last week I talked about DNA methylation, and there we saw that DNA methyltransferase is the driver there. And the erasers are the heads, 10-11 translocation proteins which can remove methyl from DNA. And today we saw that also histones can be modified, and that is mediated by histone acetyltransferase that writes and that puts acetyl groups on histones, or histone deacetylases that remove acetyl groups from histones. Methylation can be mediated by lysine methyltransferase or lysine demethylases. It is specifically on the lysine group, of course, although also arginine can be methylated. That's not shown here on this slide. And then you have readers. You have lots of proteins that interact with that modified chromatin. Like the bromodomain containing proteins, the chromodomain domain containing proteins, and so on. So a few of them are shown here. If you would come across them in literature, you can, based on their domain that they contain, we can predict with which mark they are interacting, and yeah. I hope you know what domains in proteins are. They are parts of proteins that have a specific function or can interact with something. These domains have, if your protein contains such a domain, then you can say, okay, very likely it will interact with a histone, a methylated histone, an acetylated histone, and so on. So this is also shown in the book in chapter 6. As always, I show this here, that if you want to work with the book, it can help you to have more background. But certainly do not study these parts here, because I have not given them in detail. Some parts are a bit too much for me. I think I go too far. So those parts certainly should not focus on. And also chapter 5, I'm not going to discuss. Now last week we talked a lot about DNA methylation, and now you could ask the question, which of the two, how do they compare with each other? And we notice that DNA methylation is a bit more stable than histone modification. As I already said, histone modifications is really dynamic response very easily to environmental cues. So it's a quick switch. Putting some regions on or off by histone modification can happen quite fast. DNA methylation is a bit more stable. It takes a bit more time to remove the methyl group or to add the methyl group. So typically histone modification comes first, and then later DNA gets methylated as well. So if you want to silence, if the genome wants to silence a certain part and want to go to heterochromatin state, it will first modify the histones and then only modify DNA. So, and that dynamic regulation makes it also very difficult to study the histone groups, because very quickly things change. So you're almost always too late to monitor, because very likely already after a few minutes it changes to make the gene active, and it changes back to the inactive state, and you sometimes just miss the moment that it was happening. In general, euchromatin is characterized, that's the easy one to work on. Histone acetylation is in high levels present in euchromatin. In a new chromatin, also, this open state, I told you H1 is the lock. H1 is uncommon in new chromatin. Low levels of H1. And I specifically say low and not absent. You will have a little bit of H1, but way less than in the little chromatin. Also, very low levels of histone methylation, at histone 3, lysine 9. But not the inverse, in fact, very high histone methylation at lysine 4. Histone 3, lysine 4 is the mark which is associated with those poised genes, ready for activation. So in new chromatin you have lots of methyl forms, but then not on lysine 9. You also have low levels of that heterochromatin protein 1. This is new chromatin. You don't have heterochromatin protein 1, a lot less. So low levels of heterochromatin 1, heterochromatin protein 1. And also low amounts of DNA methylation. So if you now want to go from euchromatin, the state that I just described, towards heterochromatin, and the genome wants to change the status of the chromatin at specific locations, then you need some steps. And as I said, histone modification is the most dynamic, typically comes first. So the first thing that will happen is the histones will be deacetylated by H steps. After that happens, SUB39H1, this is in humans that we are describing this process here. SUB39H1 is a human lysine methyltransferase. It will 3-methylate H3K9. And the SUB39H1 has a partner. It's always very closely connected to HP1. Those two here bind to each other and work together. So when those methyl groups are added, HP1 that likes to bind to that methylated H3K9 will bind to the chromatin. If that happens, DNA methyltransferase will come across and will stabilize the silencing by methylating the DNA. So this is a process that happens. You see it happening here. All those nucleosomes continuously, one by one. You see it happening. It spreads, let's say. It goes over the chromatin. Those two partners, the HP1 and SUB39, will do that at a specific location and will heterochromatinize. Is that a verb? Will convert the chromatin into heterochromatin. And they will continuously do that until they have some kind of stop. And that stop is a so-called insulator or boundary element. So in our genome we have elements that can stop spread of heterochromatin, which we call insulated or boundary elements. An example of where this happens, during embryogenesis, some regions of our genome, so some transcription factors, some genes are only needed at specific moments in embryogenesis. They only need to be active then. So pluripotency transcription factors like OCT4 and NANO are well known to be active very early, very early in an embryogenesis. Then they are needed, but then when they are not needed anymore, they need to be silenced, so that later in our development, the pluripotency is of course not active anymore. So to silence them, they are in our body in heterochromatin state in most of the cells, not pluripotent, of course. But during embryogenesis only, they are in euchromatin state, and then they're heterochromatinized when they are not needed anymore in later developmental stages. And that involves all these steps that I described here. So first, acetylation, deacetylation, of the histones, 3-methylation at H3K9, heterochromatin protein 1 binds, DNA gets methylated, and you get heterochromatin. So, little game. It's not difficult, don't worry. Based on what I said now, you have open chromatin and euchromatin, and you have condensed chromatin or heterochromatin. Let's go over different things here, and then you point in the direction. Will it go to heterochromatin or will it go to euchromatin? If you get more histone acetylation, in which direction will the arrow point? Will you get more euchromatin or heterochromatin? Yeah, okay. You're all looking at each other, but I see most people are indeed pointing in that direction. Histone acetylation, euchromatin. DNA methylation, and it's the common rule that I'm talking about. What will be the likely effect? DNA methylation. I would also say heterochromatin has lots of DNA methylation. But there are exceptions. So it's getting harder and harder. Histone methylation. You did the right thing. I don't know. It really depends. You cannot point in any direction. DNA demethylation. I would also point more in that direction. Typically in euchromatin you have low amounts of DNA methylation. But this is a hard one, for some of them you really are like, but there are so many exceptions to that. We have some common rules, like for acetylation, that's the easiest one, acetylation. But for the other ones, it really depends on their location in the genome and even in the gene. It depends on the organism as well. And so, yeah, it makes it hard. So the epigenome, in general, consists of multiple layers. And we have discussed already a few of those layers now. At level 1, we have discussed DNA methylation, and in repressed state you often have high levels of DNA methylation, while in active genes you have lower amounts of DNA methylation. The nucleosome organization, I will talk about that next week, but histone modifications we have discussed today. The methyl groups that are added on the N-terminal tails, or the acetyl groups. And of course, this is also helping the epigenome, or structuring the epigenome, let's say, because this histone code attracts certain proteins, like those chromodomain proteins, HP1 that I described, that are helping to compact the DNA. So those proteins that bind to the modified histones, in fact, are another layer of epigenome. And here you will have lots of those proteins, like HP1, lots of HP1 in repressed state, while less HP1 in the active state, so that, of course, this makes the DNA more accessible, all this loose chromatin thing for RNA polymerase. The nuclear organization is, where is that chromatin within the nucleus also affects itself. And that's also something that I will explain. When we study the epigenome, there are some correlations that we can observe. So DNA methylation is the most commonly studied epigenetic layer. In epigenetic studies, people typically work on DNA methylation, cytosine methylation. And that is because we have a lot of techniques to study that. I described those techniques last week: whole genome bisulfite sequencing, MIDIP sequencing, bisulfite PCR, and so on. So we know quite a lot about DNA methylation in many genomes, in many organisms. what we typically see is transposable elements, repetitive elements, are hypermethylated. While there are low levels of methylation in promoters of CP with CpG islands. Also, some methylation sensitive transcription factors like CTCF, I will talk about that one later, also typically bind to that lowly methylated region. We often see an inverse correlation between DNA methylation of promoter and gene expression. So low DNA methylation in the promoter, in those enhancer regions, leads to high expression. The word often is important, not always. But commonly. However, high levels of DNA methylation are found in active genes in their gene body. So the location where the DNA is methylated within the gene even influences. And we often also see that in disease, we have a dysregulation of the epigenome. In healthy cells, compared to diseased cells, there are major changes in the epigenome. And some people say cancer is a genetic disease. I say no, cancer is an epigenetic disease. Okay, some mutations, of course, are causing cancer, but you see way more epigenetic changes than you will find genetic changes in tumor cells. In tumor cells, and also in other diseases, we see very often changes in the epigenome, but this is the example mainly of tumor cells, where you clearly see that repetitive sequences in those tumor cells are unmethylated. While they should be methylated to keep the genome stable, in the diseased cells, they are unmethylated. And that is, of course, a very high risk for the genome, because now these repetitive sequences can repeat themselves. If they are transposable elements, they even start jumping around in the genome, maybe causing a mutation somewhere else. So this is very bad for the stability of the genome. Other observations from comparisons of the epigenome between healthy and diseased cells, and I'm talking here about DNA methylation, that's where we have most data on. Some gene bodies that are normally methylated suddenly become unmethylated. If that happens in a tumor suppressor gene, it can have dramatic effects. Or in an oncogene that activates tumor formation can have dramatic effects, of course. And some CpG islands, and this is of course bad for the tumor suppressor gene. If the CpG island of a tumor suppressor gene gets methylated, of course you get tumor formation. And this is happening in oncogenes, this is happening in tumor suppressor genes, typically in tumor cells. So we know those things from the DNA methylation across the genome in healthy versus diseased cells. For histone code, it's a bit less clear, let's say, but also there we already know that there are some disturbances in histone modification patterns that are only seen in cancer cells. All the ones for which there is some evidence are shown here with the lightning bolts. Histone 3 is the most commonly studied one. I don't know why. It's probably because it has lots of modifications. For some reason, we tend to focus on histone 3. So for that one, already quite a lot of studies have been done, and there is evidence that in cancer cells there is a shift in the histone code, so that there is more or less of that mark in those cells. Here for histone 4, also 2 marks. This is just an illustration, you don't have to study that, it's just to show you that there is a role also for epigenetic changes, a big role in fact for epigenetic changes in cancer formation, and that more and more we are looking at the epigenome when we are studying that disease. Okay. Are there questions?
about the heterochromatinization? Will the self be like permanently silent, or is just like pose? Will it turn like permanently into a
No,
chromatin, or
it can be facultative heterochromatin. So we have facultative and constitutive heterochromatin. Some parts of the genome are in constitutive heterochromatin, are always heterochromatin, like the centromeres, the telomeres. And then you have parts that can go back and forth, either euchromatin or heterochromatin.
So even the process, the heterochromatinization, will depends on the region that is happening.
Yes, yes. So some regions will in some cells be in heterochromatin and in others be in euchromatin. And that determines which tissue identity they have. So if you would compare the epigenome between all the cells in our body, it will be different. Every cell in our body, well, not within a tissue, it will be very similar. But if you compare my skin cell with a liver cell, for example, the epigenome will be very different. Because in the liver cell, some other genes are active than in the skin cells. So other genes are in euchromatin state than in skin cells. And during development, those marks are set to determine, and I will talk about that in one of the next classes, about during development how epigenetic marks are set to control which genes are needed at that moment in development and which genes are not needed. And then they transmit that epigenome to the daughter cells so that the daughter cells again have the same identity, but they can perform the same function. So every skin cell will give rise to another skin cell. If it duplicates, you will get two skin cells. You cannot suddenly go from a skin cell without tricks. You can do that, but in a lab you can do that. But in normal conditions, your skin cell will give rise to two skin cells again if it duplicates. It's doubled. Okay, other questions? If not, we have a short break here.